Hello everyone, we are Group 9. Welcome to our presentation on the basic application of a bioreactor in the fermentation process. Have you ever wondered how products like antibiotics, yogurt, vaccines, enzymes, or even biofuel are produced on a large scale? The answer often involves a bioreactor. In this video, we will explore the important components of a bioreactor, understand how each part functions, and analyze the relationship between microbial growth rate, product yield, and glucose consumption over time. Now let's dive into the external hardware involved in the air supply system. Oxygen is extremely important in aerobic fermentation because microorganisms need oxygen to generate energy for growth and metabolism. First, we have the rotameter, also known as the flow meter. Its function is to regulate and measure the amount of air entering the bioreactor. By controlling airflow, the operator can ensure that microorganisms receive enough oxygen without causing excessive foaming or turbulence. Next is the air compressor or air pump. This device generates pressurized air and pushes it into the bioreactor system. Without this component, oxygen transfer into the fermentation broth would be inefficient. Together, the air compressor and rotameter help maintain proper aeration, which is one of the most critical factors in successful fermentation. At the top of the vessel is the vessel head plate, which acts like the main connection hub of the bioreactor. It contains several ports for sensor, tubing, fitting lines, and motor attachments. Motor shaft coupling and motor mounting bracket are the structural and mechanical support components of the bioreactor. Connected to the motor is the motor shaft coupling. This component transfers rotational energy from the motor to the shaft inside the vessel. In simple terms, it allows the motor to drive the mixing system efficiently. The motor mounting brackets provides physical support for the motor. Since the motor continuously rotates during operation, strong support is necessary to ensure stable agitation and reduce vibration. Without this support system, proper mixing and oxygen distribution inside the bioreactor would not be possible. Now let's look at the main body of the bioreactor. Before that, the motor drive system is located at the top of the reactor and it provides the mechanical power to turn the internal agitator or impeller. The vessel is commonly made from borosilicate glass because it is highly resistant to heat and chemicals. The transparent design also allows the operator to observe the fermentation process directly, such as checking mixing patterns or foam formation. Some vessels are double wall for water circulation to control temperature, while others use a heating blanket wrapped around the outside. Interestingly, the vessel is not filled completely with liquid. Usually only about 70 to 80% of the total volume is used. The remaining empty space, called the head space, allows proper gas exchange and prevents overflow caused by foam. The control panel acts as the brain of the bioreactor. It continuously monitors important parameters such as pH, temperature, and dissolved oxygen. Operators can also adjust settings through this panel to maintain ideal fermentation conditions. This automation improves efficiency, accuracy, and consistency during the fermentation process. Inside the bioreactor, several important components work together to ensure efficient mixing and oxygen transfer. First is the shaft, which connects the motor to the impeller. The shaft transfers rotational force from the motor into the liquid medium. Attached to the shaft is the impeller, also known as the agitator. As the impeller rotates, it mixes the fermentation broth evenly. This helps distribute nutrients, maintain uniform temperature, and prevent microorganisms from settling at the bottom. The rotating impeller also breaks large air bubbles into smaller bubbles. Smaller bubbles provide a larger surface area for oxygen transfer, making aeration more efficient. Another component is the sparger. The sparger introduces air into the broth in the form of bubbles. It works closely with the impeller to improve oxygen distribution throughout the vessel. Finally, we have the buffers, which are metal strips attached to the inner wall of the vessel. Their function is to prevent vortex formation and improve turbulent mixing. Without buffer, the liquid will simply swirl around without proper mixing efficiency. As you can see, there are two different colors of circles shown in the picture. For the green one is the pH sensor and the red one is the PO2 sensor. The pH sensor measures the acidity or alkalinity of the medium to ensure optimal microbial growth. The dissolved oxygen sensor monitors the concentration of oxygen dissolved in the fermentation broth. Here shows the peristaltic pumps. These pumps are used to add substances such as acid, base, nutrients, or antiform solutions into the bioreactor in a controlled manner. The acid-base sensor works in conjunction with the pH sensor to trigger the addition of acid or base to maintain a stable pH level. However, for the temperature probes, it measures the internal temperature of the culture. Since microorganisms are sensitive to temperature changes, the system automatically activates heating or cooling mechanisms when necessary. After sterilization, the bioreactor is carefully reassembled. Sensors, tubing, and other components are connected properly to ensure safe operation. Important parameters such as temperature, pH, agitation speed, and airflow are configured through the control panel before the fermentation process starts. Before fermentation begins, the bioreactor must undergo sterilization to eliminate unwanted microorganisms that could contaminate the culture. One common sterilization method is autoclaving, which uses high-pressure steam at high temperature. This process effectively destroys bacteria, fungi, and spores that may interfere with fermentation. For analysis of sample concentration changes during fermentation, we measure absorbance at 540 nm. The NS method was used to determine glucose concentration and consumption. GC analysis was used to determine the ethanol production during fermentation.
The graph above shows log phase of biomass against time for two types of bioreactors. Figure 2 shows ethanol use against time for 5 grams and 20 grams yeast extracts in different bioreactors. This figure shows glucose consumption against time for 5 grams and 20 grams yeast extracts in different bioreactors. Based on the result of 10, bioreactor A containing 5 grams of yeast extract showed a higher microbial growth rate compared to bioreactor B, which containing 20 grams of yeast extracts. This indicates that the microorganism in bioreactor A adapted and multiplied more efficiently during the log phase. The higher growth rate in bioreactor A may be due to a more balanced nutrients concentration. Although yeast extract provides nitrogen, vitamin, and amino acid required for microbial growth, excessive yeast extracts in bioreactor B may have caused nutrient imbalance or osmotic stress that reduce microbial activities. In addition, high nutrient concentration can sometimes promote byproduct formation instead of active cell growth. Furthermore, better oxygen transfer and mixing efficiencies may have contributed to a higher growth rate in bioreactor A. Efficient agitation and aeration improve dissolved oxygen distribution, allowing microorganisms to grow under optimal conditions. From Figure 2, ethanol production increased over time in both bioreactors. However, the ethanol use pattern differs between the two systems. Bioreactor B produced ethanol more steadily and reached a higher overall use compared to bioreactor A. This suggests that although microbial growth was slower in bioreactor B, more substrate was directed towards ethanol production rather than biomass formation. The additional nutrients supplied by higher yeast extract concentration may have enhanced enzyme activity involved in fermentation pathway, leading to improved ethanol synthesis. Meanwhile, bioreactor A showed faster cell growth but lower ethanol accumulation, indicating that more glucose was utilized for biomass production instead of metabolic formation. From Figure 3, the glucose concentration decreased with fermentation time in both bioreactors, confirming that glucose was consumed as the main carbon and energy source for microbial metabolism. Bioreactor A demonstrated faster glucose consumption during the early fermentation stage, which corresponds with its higher microbial growth rate. The microorganism rapidly utilized glucose for cell division and metabolic activities. In contrast, glucose consumption in bioreactor B occurred at a slower but more consistent rate. This may indicate that the cells were metabolizing glucose more efficiently towards ethanol production rather than rapid biomass growth. This table compares the performance of bioreactor A and bioreactor B during the fermentation process. Bioreactor A show a higher microbial growth rate and faster glucose consumption. This indicates that the microorganism utilize glucose rapidly for cell growth and metabolic activities. On the other hand, bioreactor B produced a higher ethanol use, although its growth rate was lower. The glucose consumption was lower but more consistent, suggesting that more glucose was converted into ethanol rather than biomass. Overall, bioreactor A favored biomass production, while bioreactor B was more effective for ethanol production. Based on the result attempts, lower yeast extract concentration was more suitable for microbial biomass growth because it showed a higher growth rate. In contrast, higher yeast extract concentration promoted higher ethanol production, indicating that the microorganism directed more metabolic activities towards product formation rather than cell growth. The fermentation performance was influenced by several factors such as nutrient concentration, oxygen transfer efficiency, and microbial metabolism. Therefore, optimization of fermentation conditions is very important in industrial applications to improve productivities, product use, and overall fermentation efficiency.